Hello, friends. The gene revolution marks a significant shift in our approach to biology and medicine. At its core lies the understanding of DNA, the molecule that carries the genetic instructions for life. DNA is composed of 4 nucleotides: adenine, A, thymine, T, cytosine, C, and guanine, G, which pair in specific ways to form the double helix structure. This sequence of nucleotides is what makes each organism unique. DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. The discovery of DNA's structure in 1953 by James Watson and Francis Crick revolutionized biology. This breakthrough laid the foundation for modern genetics, enabling scientists to decode the genetic information that governs the development and functioning of all living organisms. Watson and Crick's model explained how DNA replicates and how hereditary information is coded on it. They also shared the Nobel Prize for Physiology or Medicine in 1962 with Maurice Wilkins, who was also crucial to the discovery. The Human Genome Project, completed in 2003, was another monumental milestone. This international research effort successfully mapped the entire human genome, revealing the sequence of all 3 billion base pairs of DNA in human chromosomes. The Human Genome Project has significantly impacted biology and medicine, allowing for advancements in personalized medicine, drug development, and the diagnosis and treatment of genetic diseases. Understanding the history leading up to these discoveries provides context for the significance of the gene revolution. Gregor Mendel's work in the 19th century on the inheritance of traits in pea plants laid the groundwork for the field of genetics. Mendel's meticulous experiments and formulation of the laws of inheritance demonstrated that traits are passed down from parents to offspring in predictable patterns. His principles of heredity were later rediscovered and expanded upon, leading to the integration of genetics with molecular biology. The 20th century saw significant advancements, including the identification of DNA as the genetic material by Oswald Avery and colleagues in 1944, and the development of the polymerase chain reaction, PCR, by Kary Mullis in 1983, which revolutionized genetic research by allowing the amplification of DNA sequences. These historical milestones highlight the progressive nature of genetic research and its foundational role in modern science. The ability to read the genetic code opened up new possibilities for understanding and manipulating genes. Genetic engineering emerged as a field that allows scientists to alter the genetic makeup of organisms. Early techniques involved the use of recombinant DNA technology, where genes from one organism could be inserted into another. This led to the creation of genetically modified organisms, GMOs, which have applications in agriculture, medicine, and industry. One of the earliest examples of genetic engineering was the production of insulin by genetically modified bacteria, which provided a more reliable and abundant source of this essential hormone for diabetes patients. This breakthrough marked the beginning of the biotech industry, showcasing the potential of genetic engineering to address complex biological problems. One of the most groundbreaking technologies in genetic engineering is CRISPR-Cas9. Discovered in the early 2010s, CRISPR allows for precise editing of the genome by targeting specific sequences of DNA. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The simplicity and efficiency of CRISPR have made it a revolutionary tool in genetics, enabling researchers to make targeted modifications with unprecedented accuracy. This technology has vast potential applications, from treating genetic disorders to enhancing crops and livestock. For instance, CRISPR has been used to develop disease-resistant plants and to engineer animals with desirable traits. The ability to edit genes with precision has opened up new possibilities for understanding gene function and developing novel therapies. To illustrate the impact of genetic engineering, consider the case of severe combined immunodeficiency, SCID, also known as bubble boy disease. Patients with SCID are born without a functional immune system, making them highly susceptible to infections. Traditional treatments involve bone marrow transplants, which are not always successful. Gene therapy has offered new hope by introducing a correct copy of the defective gene into the patient's own cells. In one notable case, a young boy named David Vetter lived in a sterile environment due to SCID. Although he passed away before gene therapy was available, modern techniques could have transformed his life. Gene therapy trials for SCID have shown promising results, enabling patients to lead normal lives without the need for isolation. Another success story is the treatment of Leber congenital amaurosis, LCA, a genetic disorder that causes severe vision loss at birth. Gene therapy has been used to deliver a functional copy of the defective gene directly to the retina. Clinical trials have demonstrated significant improvements in vision for patients treated with this therapy. These cases highlight the transformative potential of gene therapy in treating previously incurable genetic disorders. Moreover, the development of gene therapy for hemophilia has shown how this technology can replace lifelong treatments with a single intervention, significantly improving patients' quality of life. To gain deeper insights into the gene revolution, we can imagine interviews with leading scientists in the field. Dr. Jennifer Doudna, one of the pioneers of CRISPR technology, could share her experiences and thoughts on the future of gene editing. She might discuss the potential for CRISPR to revolutionize medicine and agriculture, as well as the ethical considerations that come with such powerful technology. Similarly, Dr. Eric Topol, a leading figure in personalized medicine, could explain how genetic research is transforming healthcare by enabling treatments tailored to individual genetic profiles. These expert perspectives provide a more comprehensive understanding of the scientific, ethical, and societal implications of the gene revolution. Hearing directly from these innovators could also inspire a new generation of scientists and researchers. Looking ahead, the possibilities of genetic engineering are both exciting and daunting. One optimistic scenario envisions a future where genetic diseases are eradicated. Scientists could use gene editing technologies to correct harmful mutations before they cause disease. This could lead to a significant reduction in the prevalence of genetic disorders and improve the quality of life for millions of people. On the other hand, the potential for creating designer babies raises ethical concerns. The ability to select for specific traits such as intelligence or physical appearance could lead to societal inequalities and ethical dilemmas about what it means to be human. Balancing the benefits and risks of genetic engineering will require careful consideration and robust ethical guidelines. Additionally, the integration of artificial intelligence with genetic research could accelerate discoveries and applications, further transforming the field. Public attitudes towards genetic modification vary widely across cultures and regions. In some countries, there is strong support for the use of genetic technologies to improve health and agriculture. For example, many Americans view GMOs positively, seeing them as a way to increase crop yields and reduce pesticide use. However, in Europe, there is more skepticism and opposition to GMOs, driven by concerns about food safety and environmental impact. Religious and ethical perspectives also play a significant role in shaping public perception. Some religious groups oppose genetic modification on the grounds that it interferes with natural processes, while others support its use to alleviate suffering and improve human health. Understanding these diverse viewpoints is crucial for developing policies that are respectful of cultural and ethical differences. Effective public engagement and transparent communication are essential to build trust and ensure informed decision-making. The development and application of genetic technologies are governed by a complex web of regulations. In the United States, the Food and Drug Administration, FDA, oversees the approval of gene therapies, ensuring they are safe and effective before they reach patients. Similarly, the European Medicines Agency, EMA, regulates the use of gene therapies in Europe. International organizations like the World Health Organization, WHO, provide guidelines and frameworks to ensure the ethical use of genetic technologies globally. These regulatory bodies face the challenge of keeping pace with rapid scientific advancements while ensuring public safety and ethical standards. Developing harmonized global standards for genetic research and applications is essential for fostering innovation while protecting individuals and society. These frameworks must also address the potential risks of gene editing, such as unintended genetic changes and ecological impacts. Beyond CRISPR-Cas9, several new genetic engineering technologies are emerging. CRISPR-Cas12 and CRISPR-Cas13 offer additional capabilities for gene editing and targeting RNA, respectively. Base editing, developed by Dr. David Liu and colleagues, allows for the precise conversion of one DNA base into another without making double-strand breaks. This technique has the potential to correct point mutations that cause many genetic diseases. Prime editing, another innovation from Dr. Liu's lab, combines the best features of CRISPR and reverse transcriptase to make even more precise edits to the genome. These advancements enhance our ability to manipulate genetic material with greater accuracy and fewer off-target effects, expanding the potential applications of genetic engineering. These technologies hold promise for treating a broader range of genetic conditions and for more complex genetic modifications. The gene revolution can be compared to other technological revolutions, such as the digital revolution, in terms of its transformative impact on society. Like the digital revolution, which brought about the widespread use of computers and the internet, the gene revolution is fundamentally changing how we understand and interact with the natural world. Both revolutions have led to significant advancements in science, technology, and medicine, and have raised important ethical and societal questions. However, while the digital revolution primarily affected information and communication, the gene revolution directly impacts the biological foundation of life itself. The lessons learned from managing the digital revolution, such as the importance of ethical considerations and regulatory frameworks, can inform our approach to the gene revolution. Both revolutions also emphasize the need for interdisciplinary collaboration to address complex challenges. To help the public understand the gene revolution, a variety of educational resources are available. For beginners, textbooks like Genetics, A Conceptual Approach by Benjamin A. Pierce provide a solid foundation in genetic principles. For more advanced learners, scientific journals such as Nature Genetics and the American Journal of Human Genetics offer the latest research findings. Online courses from platforms like Coursera and edX provide accessible learning opportunities, with courses like Introduction to Genetics and Evolution from Duke University and Genomics from the University of Illinois. Reputable websites including the National Human Genome Research Institute, NHGRI, and the World Health Organization, WHO, offer comprehensive information on genetic research and its applications. These resources can help demystify genetics and empower individuals to engage with the science and its implications. The applications of genetic engineering extend far beyond medicine. In agriculture, genetically modified crops have been developed to increase yields, resist pests, and tolerate extreme weather conditions. For example, golden rice, engineered to produce beta-carotene, addresses vitamin A deficiency in developing countries. In industry, genetically modified microorganisms are used to produce biofuels, biodegradable plastics, and other sustainable materials. These innovations help reduce reliance on fossil fuels and decrease environmental pollution. Additionally, synthetic biology is being used to create novel organisms with specific functions, such as bacteria that can break down pollutants or produce pharmaceuticals. These real-world applications demonstrate the broad potential of genetic engineering to address global challenges. The continued development of these technologies could lead to more sustainable and efficient production methods across various sectors. As we continue to explore the potential of the gene revolution, it is essential to engage in public dialogue and education about genetic science. Understanding the basics of genetics and the implications of genetic technologies is crucial for making informed decisions about their use. Public awareness and education can help to ensure that the benefits of the gene revolution are realized while addressing the ethical, social, and environmental challenges that it presents. Educational initiatives should aim to make genetics accessible to a broad audience, providing clear and accurate information about the science and its applications. Outreach programs, public lectures, and interactive exhibits can help to engage the public and foster a deeper understanding of genetics. Encouraging the inclusion of genetics education in school curricula can also help to build a foundation of knowledge for future generations. The gene revolution is a transformative period in the history of science and medicine. Advances in genetic research are unlocking the secrets of life, offering new ways to treat diseases, improve agriculture, and understand our evolutionary history. However, these advancements also come with significant ethical and social considerations that must be addressed. By fostering an informed and engaged public, we can navigate the challenges and opportunities of the gene revolution to create a better future for all. Thank you for joining us on this journey through genetic engineering. Please like, comment, and share the video, and please do not forget to subscribe for more videos.